Welcome everybody. Good to see a nearly full house. We've got a couple of seats empty. I think a couple of people will pop in during the presentation, but I'm sure you all won't mind that. Thank you very much for joining us all today. I would like to open today by acknowledging the traditional owners of the land, the Whadjuk Noongar people, and I would like to pay my respects to the elders past, present and emerging. I'd like to also acknowledge any Aboriginal and Torres Strait Islanders with us today, and to thank our Indigenous culture for allowing us to continue to enjoy this beautiful country. For me today, I'm just going to do a quick little update so you're all aware. I know majority of people here today are community members with us that either live with a neuromuscular condition or care for someone with a neuromuscular condition. So if you're not familiar with all of our events, we have a whole heap of events coming up in the next while, so we thought we'd just give you a quick prompt. We've also got some flyers here which you're welcome to take there's some on your desk as well. On the tables, which you're welcome to take with you as well and pop them on the fridge. So firstly, we have female carers, which is on the 29th of June, which is in a couple of weeks' time. That's brekkie for all the female carers. That's in Leederville. The following week, we have male carers event, which is anyone. I don't think I need to explain myself what male carers events are, or female carers. So that is at Flight Club, and that is always lots of fun. Not that I've ever been to one, but Holly, who does work with us, always reports it's a big hoot and everybody enjoys it. YASAG, for those who are not familiar with YASAG are our young adults social club. So anyone that is over the age of 25-ish. And do we have a top end, Piper, or for YASAG, do we have a top end age? It says it on there. No, just age-wise.
Oh, no.
Okay. We have a floor but no ceiling. So that is end of July, Sunday the 28th of July. And then we also have a Conversations and Connections evening. Is it an evening? Yes, afternoon evening, which is about manual handling. It's just a refresher course and also some discussion and conversation. Final event, that's just to let you know, we haven't done a flyer yet for this one because this is still a way out, but on the 10th of September we have a will writing session which will be here at the niche, and we'll have that'll be presented by an external consultant. So that's sort of just a little bit about what's coming up. Something new that we just wanted to let you all know is this year we've started an adolescent social group. So back in 2015 there was a gap in social support for young adults, and we started our young adult social group, but those young adults have all progressed to into their 20s and 30s, and they're still part of YASAG. So we've decided to create a group which are just an adolescent group. So basically that's for if you have any adolescents from the age of leaving school up until 25, that group is there to support them. So it's about peer support, it's about networking and learning, and ideally what we will do is we will then introduce those older group members into the young adults group at the right time for them. So it's all a very caring, supportive environment, lots of fun. So if you do have any adolescents or you know of, please put them in touch. System navigation, I'm quite sure most of you are very familiar. If you've been reading our newsletters, you would be aware that last year it's been under development, but we launched our system navigation program in May last year. It's a program that's funded by the Department of Health as a trial. The aim for this program would be to develop a navigation program for people living with rare diseases. So we've actually transitioned now to phase 2, which starts June this year, which is the next phase of that. That would be to introducing other conditions and patients living with other rare diseases to that navigation support. So basically what it is, it's you have your own navigator who's there to support you through any, I guess, struggles that you're having in and around your condition. So consider it something that's very vast and broad. It's not a one-size-fits all. It's very specific to individual needs. It basically is trying to take the stress and pressure of you in managing your condition journey and where you're at on your journey. If you're confused about where to access funding or where to access different services, or if there's you're struggling with anything within the health system, that the aim of the navigator is to provide individual support to you. And I'm very proud to say that our one navigator, who is Josh, he didn't know I was going to do this, but Josh is with us today. So Josh, we have two navigators, and Josh is one of the navigators. So Josh will work with you over a period of time to develop an action plan where you'll have some priorities which you'll work through, and then either Josh or Sarah will guide you along that path over the next year, or however long you need to be part of it. If you're already a member with Neuromuscular WA and you haven't accessed it or asked about it, please do, because it can definitely help you better access everything that you need to. To get involved with system navigation, there's a link on our website, or you can just reach out to email our system navigator, which I think is that just say system nav? Yes, at neuromuscularwa.org.au. We can provide a copy of this to you if you do want via email afterwards. So I think that's pretty much it for us and what I'm going to update you. But the main reason you are all here tonight is specifically to listen to two wonderful presenters. Their bios are this long. They're pretty famous. And so I'll just introduce both of them. What we'll do is we'll firstly have a presentation from Gina, and then we'll have a little break, and then we'll have a presentation from Rhonda. So I am going to read through Rhonda's bio, both bios, sorry, but Gina first. Gina is an associate professor, so Gina Ravenscroft. She received her PhD from the University of WA in 2009. Gina has held 3 consecutive highly competitive NHMRC fellowships and currently holds an NHMRC Emerging Leadership 2 fellowship. Gina is the Honorary Patricia Vern Kalis Fellow, and it's supported by the Kalis family. In 2020, Gina established her Rare Disease Genetics and Functional Genomics group at the Harry Perkins Institute. Her research focuses on rare genetic diseases, particularly neurogenetic diseases in babies and children. Gina has identified over 20 novel human disease genes and works with an extensive network of global collaborators on gene discovery projects. We did have a joke in the office today that your name was Gina, and actually all genes are named after you, which I'm sure you've heard many, many times before. Gina is recognized as a world leader in fetal akinesis and congenital myopathies, and was named the Young Biologist of the Year and Australian Institute of Policy and Science Young Tall Poppy in 2016. Gina is on the executive board of the World Muscle Society, chair of the WMS Scientific Programs Committee. She's a member of the scientific advisory boards for FSHD Global Research Foundation and the Foundation Building Strength for Nemaline Myopathy. So as you can see, when I said she's pretty famous and she's pretty accredited, she certainly is, and we are extremely lucky to have you here today, Gina. So welcome, and thank you so much for your time.
So thank you very much for the introduction and for the opportunity to come and meet with you at Neuromuscular WA and to meet with the community. I work at UWA and at the Harry Perkins Institute for Medical Research and lead a small team there focused on rare genetic diseases. and in particular neuromuscular diseases. So I guess that's why we're here. I've worked in the area since 2005 when I started my PhD with Professor Nigel Lang and Dr. Kristen Nowak, who some of you will know. We were then at Weymouth, which was just in B Block, which is now the Harry Perkins, and I've basically never left. And I hope that I can show you that WA in Australia is a great place to do human genetics research and neuromuscular disease research, and why Perth has played such an important role on the global stage in terms of these diseases. So there are over 7,000 different rare diseases, and the zebra is a symbol for rare disease because medical students are often taught to think of horses when they hear hooves, to always consider the most common thing first. And I guess that this sort of bias in training likely contributes to some of that diagnostic odyssey that many rare disease patients may experience, and that some of you may have experienced also. More than 80% of rare diseases are due to genetic defects, and so that's why we have labs here, but also all around the world, looking at genetics of rare diseases. And the research into genetic neuromuscular diseases is really an area of excellence in WA because of the many great leaders in this space some decades ago. The late Professor Byron Koukoulas, also Frank Mastaglia, and Patricia Verne-Kalis, who was doing genetic counselling here in WA muscular dystrophy families and doing predictive testing even before any of the genes were known. And I don't think it's really recognised how much Patricia actually did in those early years before we even had genetic diagnostics for these conditions. Neuromuscular diseases can present across the lifespan, from in utero and then in kids to later adult life. Over 40 novel human disease genes have been identified by the Perth group across the last 3 decades, and many of these have been in the last 10 years, aided by next generation sequencing. And I guess when we think about neuromuscular diseases, there's 500, 600 genes that are known in total. So for Perth, the most isolated capital city in the world, to have contributed or led, roughly 10% of those is really something that we should be really proud of, and speaks to the legacy of great research that has come from essentially this site, where ANRI used to be, and Weymouth, and now the Perkins and Perrin Institute. But despite all the advances over the years, and we know of hundreds of genes now, more than 50% of patients remain without a genetic diagnosis despite sort of gold standard diagnostic testing. So if you go to your neurologist and they send off a request form and a blood sample to have your DNA tested, only about 25 to 50% of patients will actually get a diagnosis from that test. And there's lots of reasons for that. We still have new genes to identify. There's variants that we identify that we can't interpret yet. So there's variants of unknown significance where we're pretty sure that's the cause of disease, but we don't have enough evidence to be confident and report it diagnostically. There's regions of the genome that are still difficult to sequence and to map and to understand. And so we and others around the world are trying to work out how to tackle those tricky parts of the genome, and we now have new technologies for that as well. So that's where the research side of things comes in. So if patients don't get a genetic diagnosis through sort of standard clinical testing, and they're interested in research, we then get access to those samples for research testing. So to give you an idea of some of the diseases included in our research, I generated this word cloud of all the diagnostic labels and phenotype descriptions that were listed against the most recent 800 samples that we've sent off for sequencing. And so you can see myopathies and neuropathies are quite prominent within the cohort, but also ataxias, dystonias, myotonias, limb-girdle muscular dystrophies, HSPs, metabolic myopathies, movement disorders, and many other things. So basically, if there's a neuromuscular disease and there's a high suspicion based on family history, or from the clinician thinks it's likely to be a genetic condition, then we would be willing to enrol a patient or the family in our research study. And we have funding from the federal government and from families and different grant organisations to do the work that we do. And currently we have a grant that Nigel Lang is the main lead for, that I run with him, but also collaborators in Melbourne and Sydney, that's specifically looking at identifying genes in later onset disorders. So for a long time, a lot of the focus was on kids and babies. It's much easier for lots of different reasons to find genes in causative genes in rare diseases, neuromuscular diseases, when it's very early onset disease. It's more difficult for later onset conditions. So we've realised and identified that there's this huge unmet need to try and provide answers for those other types of conditions. And so we're part of a four-year grant to look at specifically at the genetics of ataxias and neuropathies, which are typically present later on in life. So before I get too much further, I wanted to share this photograph taken at our recent lab retreat at the Perth Zoo. This shows both the rare disease group that I lead and then Nigel Lang's preventative genetics group that works on prevention but also therapies, and we work very closely together. And the work we do is very much a team effort. So I just wanted to highlight, oh, you're not going to see. Can you see my pointer? A little bit. Carolyn works on a cohort of ataxia patients. She's doing her PhD, and she's doing her PhD jointly between the research lab and PathWest. So that anything we identify in the research lab or new technologies to identify different types of mutations can then be implemented in the diagnostic setting in a really sort of quick time frame, because there's that synergy already from the beginning. Jevon is currently in Germany visiting the Embl labs there. He's working on a cohort of patients with neuropathies. Leon is working on a cohort of patients with myopathies, the primary muscle diseases. And then Kiara has recently started a PhD with the team. She's worked with us for a few years already, more in the computational side of things, trying to get the most out of the sequencing data using new computational tools. And she's given herself a really challenging project to trying to solve the unsolvables. So we have lots of families where we've worked on them for many years and they still haunt us in a way, because the cause of their disease must be somewhere in that genetic data, but we just haven't been able to find it yet. So we're hoping with the new technologies and new computational tools that we can do that for some families. Mridul Johari joined the team about 18 months ago from Helsinki, and he has an interest in distal myopathies, protein aggregate myopathies, and also IBM. So perhaps at a future meeting he might be interesting to get in to speak. And Audrey Rick is our research genetic counsellor, and she's really critical to making the whole thing work. So she liaises with the clinicians and then contacts families to talk about the research, organise consent, organise blood samples, and then once all the boxes are ticked, make sure that the samples get queued to be sent off for sequencing so that that all happens seamlessly. So those are some of the key people sort of involved in the work that I'm going to share with you today. And these are the key clinical colleagues who refer patients for our research in WA. We work very closely with Philippa Lamont, with Marilee Needham, Wayne Yao, who's at the Perrin Institute, focused with movement disorders, and Catherine Ashton, who works with Philippa but also with Marilee at Fiona Stanley Hospital. We also collaborate closely, as you might expect, with Genetic Services of WA and also with the different pathology services. If patients have had a muscle biopsy, for example, that's often really useful for our studies as well. So this was a photograph I took Monday lunchtime. We have a Zoom meeting every Monday between key staff in the research lab. Mark Davis, who runs the diagnostic neurogenetics unit at PathWest, runs the diagnostic lab, some of his staff, Nigel, Philippa Lamont, Wayne Yao and Catherine, so the neurologists, and we talk about the patients that are coming into the clinic for that week, talk about any results, we talk about any interesting variants that the diagnostic lab have identified, how the research and diagnostic lab might be able to work together to get a variant over the line, to be able to classify it as pathogenic if it's sort of not quite meeting the very strict criteria they have for diagnostic reporting of variants. And then any patients where the diagnostic testing hasn't been informative, we then discuss whether they might be eligible for research testing. So these sorts of meetings are now called multidisciplinary team meetings, and it's sort of a buzzword, but Nigel and Philippa and PathWest have been doing these meetings for, 25 years, since before MDTs were kind of a concept. And these are really great meetings that I think really benefit all of us, but also the patients here in WA in terms of trying to get as many diagnoses as we can. The field's highly collaborative, as you can see from some of the examples I'm about to share, and we've co-published with 1,300 different researchers and clinicians from across 40 countries on 6 continents. So for little Perth on the far end of the universe, we're doing really well in terms of working at the global level and at the kind of forefront of where genetics is going in for neuromuscular diseases. So a very brief crash course in your genome. Our DNA is made up of around 3 billion base pairs, and it's only a 4-letter code of A, C, T's and G's. Nigel once spoke to a typist in the days that typists were still around and worked out how fast they typed and came up with the idea that writing out your genetic code would take a pretty good typist 27 years of non-stop typing. So when we're trying to find a mutation or a spelling mistake in the DNA code, it's like looking for one spelling error, one typing error in 27 years of typing. We all have many variants in our DNA code, many thousands of changes. That's just part of natural variation. And so a challenge for us when we look at the sequencing data is to work out in a given individual which of the variants are just benign, part of their unique variation that makes us human, and what is the likely disease-causing mutation. So many of the first neuromuscular disease genes were identified using large families and linkage or mapping approaches to pinpoint the likely causative genes. So this shows a large family tree, or a pedigree we call it. The squares are male individuals, the circles are female individuals, and so you can see this particular male individual, he's affected, the symbols are coloured in. So he's affected, he's had 2 daughters, and he's passed the disease on to this daughter, but not the other one. And so we can look at the DNA in these large families and look at which regions of the genome are inherited or the same between all the affected individuals, but not in the unaffected relatives. And then we can look at we can work out which regions of the chromosome are the same in affected individuals, but not in the unaffected individuals, and then sort of say, well, we think the variant in this family is on this particular region of a particular chromosome. And that's how, and then they go through the process of saying, well, thanks to the Human Genome Project, we sort of know where all the genes are in the genome, and say, well, within this region of a particular chromosome, which genes are within that region, and which of them might have a function in muscle if it's a myopathy, or which of them might have a function in a peripheral nerve if you've got a CMT, family with CMT. So that was sort of the process that people used. And that was something that Professor Nigel Lang did in the early 1990s. They had this large South Australian family with the diagnosis of Mennelin myopathy, which is a type of congenital muscle disease. They mapped the disease to a region at chromosome 1, and the Human Genome Project was sort of still getting worked out, and they sequenced a few genes within that region. It wasn't any of those, and then the map kind of got updated, and there was a new muscle gene now within that region, and they then sequenced that region, and that was the tropomyosin gene. And they identified a mutation in the tropomyosin gene that was the cause of Mennelin myopathy in this family. And we knew from sort of functional work that tropomyosin is a really important protein for muscle contraction. So then Nigel started receiving DNA from all around Australia, from different families, and all around the world from families and from clinicians that work with these families with these genetically undiagnosed neuromuscular conditions. And that's part of the reason that we have this really rich legacy of research here in Western Australia, is that sort of it got recognised as a centre that was active in this space, and so samples came and they found more things, and so more samples came, and so it sort of grew from that. Over the next 30 years or so, more than 10 other genes were identified to cause Mennelin myopathy. Most of these discoveries were either led by the Perth team or included the Perth team because of all the samples that were sent here. And you can see from the huge list of authors, so each of those titles is a particular paper. It's a new gene associated with this particular type of muscle disease, and all of the names, all of the people involved from all over the world with each of those studies. So I like to tell Nigel that he used to work in the dark ages, where you took your little Sanger sequencing and you sequence one little bit of the DNA, sequentially, one at a time. You did this exon and then that exon of the gene and then the next exon. And it took a lot of time, and you couldn't, depending on how big the gene was, you couldn't sequence the whole gene. So they used to sequence just chunks of genes where they knew mutations were really concentrated in a hot spot, and there wasn't any way to comprehensively sort of sequence lots of genes at one time. And then in the early 2010s, next generation sequencing became more available. This is a process where you fragment DNA and then you sequence it in a massively parallel way. So instead of doing one exon at a time, you just sequence the entire genome. And so while the Human Genome Project took 20 years and around $3 billion, we can now sequence an entire human genome for $1,000 in a day or two. So we can generate the sequencing data really easily, and it's relatively inexpensive nowadays compared to even, say, a muscle biopsy and some of the other procedures that we have to try and get a diagnosis. And then the trick is to try and work out, we don't have a linkage region, the entire genome. The variant is somewhere in your genome, and then we have to sift through all of the thousands of variants to work out which one or two variants in your genome is the cause of the disease. And so one way that we adopted next generation sequencing here in Perth was that because of this synergy between PathWest and the research lab, we worked really closely together and developed targeted gene panels to sequence all of what the known genes in the early 2010s for neuromuscular conditions. So we implemented that into diagnostics through these sort of targeted panels, and to date, PathWest has sequenced over 8,000 patients on these panels from all around Australia and New Zealand. And it's sort of now a national referral centre for the genetic diagnostics of neuromuscular conditions. And you'll see from this table that around 25%, so 1 in 4 patients, get a diagnosis straight off the panel. You can also see, if you look at the different subtypes of disease, that this really varies across subtypes. So for example, congenital muscular dystrophy presents in birth. These are congenital, as the name implies, dystrophies. About 60% of patients will get a diagnosis off the panel, whereas something like ataxias, which are later onset, only 15% of patients get a diagnosis off the panel. And so the patients that are then unsolved off from the panel are then enriched for discovery of new genetic causes of disease, and those are often the ones that get recruited into research. So over the past 10 or so years, we've identified and characterised lots of genes, as was said in the introduction, and each of these discoveries really means that another cohort of patients here in Australia, but around the world, also receive an accurate molecular diagnosis. It's included in panels and clinical testing all over the world. So if a patient now presents with a myopathy, all of these genes will be considered in addition to what was known and what's been identified in other parts of the world. And the clinical management can then be tailored to their specific condition. They can reach out to other families who have that same precise genetic condition, and there can be screening of relatives too, if appropriate, and if relatives are interested in finding out. So in bold, I've highlighted all the discoveries that involve early onset conditions. For a number of reasons, as I've said, it's much easier to find genes in the paediatric setting than in the sort of later onset diseases. And often we refer to these as sort of the low-hanging fruit in human genetics. They're kind of the easy things to get at. But I'm now going to share with you a couple of examples from discoveries we've made in neuromuscular diseases in adults. So the first is this particular family. We received the first the first DNA sample from this family. You identified yourself, I wasn't going to. In 1992, we only found the gene, I think, in 2017. So it was a 25-year diagnostic odyssey. There was a diagnosis of peripheral neuropathy. And what we did is we took DNA from 5 affected individuals in this family and 2 unaffected individuals, and we didn't quite have enough individuals to do linkage. I won't go into the maths, but we just decided, well, we'll look across all of the variants we identify, and we look for variants that are present in the 5 people that do have CMT, but not present in the 2 people that don't have CMT. And when we did that, there were only 6 genes that had coding variants. So coding variants are variants in genes that make a change in the protein. So genes encode proteins. Mostly, we still think protein coding changes cause disease. So we were most interested in things that change proteins. So only 6 genes had variants that altered protein sequences, and 5 of those variants were present in population-controlled databases. So over the last few years, there's been a massive effort all over the world to aggregate all the sequencing data people have generated. And they've sequenced lots of healthy individuals and then if you have a variant in a whole bunch of healthy people that don't have CMT, well, it's unlikely that that variant causes CMT. So we then filtered out all the variants that were present in people in the population that didn't have CMT, and we were left with one gene, and it was this gene ATP1A1. And this is what, I don't know if you can, it's a bit small on your screen, but this is what that sequencing typing error looks like. So we have a reference sequence here of A, C, T's and G's, and at this particular region of the genome, we have two changes next to each other. So someone made a double spelling mistake, and it changed a codon from a TGA to a TTT. And that was enough to change the protein, the amino acid sequence at position 601 in the ATP1A1 protein. So we were really excited about this because the ATP1A genes belong to a family of sodium, potassium, ion pumps, and we know that those are really important for function of neuronal tissue. Variants in two other related genes have been associated with familial migraines and with hemiplegia of childhood and with dystonia. So we had this link between this group of genes and proteins and sort of disorders of the nervous system. We also did we go on, our version of Google's called PubMed. We went on to PubMed, put the gene in, and we identified this paper, which we refer to quite a lot in our work, where they'd done proteomics. So they'd taken peripheral nerve myelin, which is really important for making sure the electrical signals get sent at speed. It wraps around the nerve and sort of helps with the conduction of the electrical signal. They'd taken the myelin from peripheral nerves and looked at all the proteins that were present in myelin and made a list. And they said, well, these are really good candidate genes for neuropathies, and ATP1A1 is one of the proteins that's important for myelin. And then the other thing we do is we look at how conserved a particular amino acid is. So the amino acid was changed at position 601. We go and look, well, in all types of species that have ATP1A1, what amino acid is present there? And it was also an aspartic acid or a D symbol. So there's the D in humans. This is a protein sequence. We now have the letters of the protein code. In all these other species, it's also a D. This is the variant in the family. So even in worms, it's a D. So that tells us that there's been this evolutionary pressure that to keep that protein sequence the same. That is really important for function. Whereas say the variant we'd identified in our family had been the same amino acid change in monkeys or cats or mice, we might have been less interested in it because there was less evidence to suggest that that particular sequence was important for function. So we then contacted Stefan Zuckner in Miami, who, if you wanted to count on one hand kind of the top neuropathy groups in the world, he'd be on that list. And we said, well, we think ATP1A1 might be a new CMT gene. And he replied straight away and he said, it's our hottest project. We have 4 big families with CMT2 with variants in ATP1A1. We have no doubt this is a new CMT gene. And he then shared his variants with us, and we shared ours with him. So ours is this 601 at position 601, and they're all really close together. So 601 means there's at least 601 amino acids in the protein. I don't know how big the protein is, but there's hundreds of them. And all 4 of the variants across these 5 families were within this very small window of ATP101. So Mark, being very practical, says at one of these Monday meetings, well, that's exon 13. Let's just sequence that one exon using the old technology in a bunch of patients and see what happens. So he did that, and he found a variant in a second family from WA. This family, the first DNA sample was received in 2002, and they also had a variant in ATP1A1. So just from sequencing that tiny little region of that gene, we identified, made a diagnosis in another family. And when we put that together, these are the 4 we knew about already. We put this one in. It's also sitting just around this. I've boxed these residues because protein people know that this motif is important. It forms a hinge. So it's important for sort of the folding of the protein. So we think that all of these variants probably impact protein folding. And this was the resulting paper. So I think all up, there were about 8 or 9 families identified. People have now studied ATP 101 variants in Drosophila or fruit flies. They've now identified variants in ATP 101 in different phenotypes. So often that happens. We identify a variant in a gene and we say, well, it causes Nemaline myopathy or it causes CMT. And then people do lots and lots of this sequencing. We're now sequencing genes in thousands of patients around the world and go, well, hang on, variants in that gene actually cause all these other phenotypes as well. So we have to accept that there's going to be this spectrum of different presentations that might be associated with variants in a given gene. And then just a week ago, we had an email from a neurologist in Newcastle saying, we've got a family with this very rare CMT. Can you put us in contact with the clinicians so that we can find out a bit more about the condition so we can tell the family as much as possible? And we did, and so that's now happening. So there's a lot of work that goes on to try and find the genes and then put it all together and prove that it's causative and then connect up the clinicians so that they understand the disease, because if there's only 10 families reported in the whole world, you know, trying to get enough information to be helpful for the next family also. And so the second example I'm going to share very quickly is that over the past, it was the identification of ABCD3 repeat expansions in a very rare myopathy called oculopharyngeal distal myopathy. And these are all the people involved in that study and all of the institutes involved, so another really big study. And again, it started with 2 Australian OPDM families. So oculopharyngeal distal myopathy means the eye muscles are involved, the muscles involved with speech and swallowing are involved. They also have facial weakness and involvement of the distal limbs, so the hands, the feet, the lower legs. So Philippa and Macarena Cabrera-Serrano, who's a neurologist, she did part of her PhD with us here in Perth because she likes summer. So she's from Seville. So she spends half of her year in Seville in summer and then has summer in Perth as well. And she's now married to an Australian, so she's kind of an honorary West Australian. So Philippa and Macarena have been looking after this family here in Perth, and there was this really large pedigree on the east coast of Australia that Professor Elston Story had sort of really well phenotyped, and Michael Fay's now the neurologist in looking after this family, and some of the individuals were seen by Garth Nicholson in Sydney as well. And this family has over 100 people in its pedigree. It's a huge, huge family tree. So we did exome sequencing on 10 affected individuals, 5 from each family. And then Sarah Beecroft, who was a PhD student with us at the time, did linkage analysis and got linkage to this region. So this is along this x-axis is sort of the chromosome 1. And we've got a peak here going up to 3, which is the magic number for LOD scores. You want a LOD score of 3. That tells you you're pretty confident that you've got good linkage. So within this region of chromosome 1, we think the variant for OPDM in these families lies. Sarah then looked at all the variants in the exome sequencing data that was within that region of chromosome 1 and couldn't find anything that she thought was likely to be the cause of their disease. And then we did, at the time, you know, I say we can now do whole genome sequencing on a person for $1,000. Probably when we did whole genome sequencing on this individual, it was $3,000 or $4,000 an individual. So we just picked one patient and did whole genome sequencing. So instead of sequencing just the exons, the coding bits of genes that make proteins, we sequenced the entire genome, and we got everything, all the bits of your genome in between your genes and in between your exons. And then we decided, Sarah again couldn't find any variants when she just looked through standard analysis of the whole genome sequencing data in that region. And then a program came along called Expansion Hunter. So this is one of these new sort of computational tools that will look for repeat expansions in your DNA. So instead of having a typo where you changed a C to a T, you insert a couple of sentences of a repetitive sequence. So we have these repetitive regions within our genome, and they're prone to expansion. So you might have a CCG or a CAG, like in Huntington's, and then you get CAG, 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 and that gets expanded. And so you have a few sentences, sometimes paragraphs worth of this repeat stuck into your genome. And we know that these expansions can cause neuromuscular conditions, and they're quite common in ataxias. And so we looked using Expansion Hunter to see if we could detect any expansions. And we did that, and it called a potential expansion in the ABCD3 gene. And so we then looked at the genome, and I think you can just see. So this is the reference sequence along here, and somewhere here there's a CCG expansion. It's usually just 7 repeats, so you've got 7 CCGs. And you can see in some of the reads we have CCG expansions indicated. So when we looked at the sequencing data, we said, well, that looks like a really real expansion. We were really excited that this was probably the cause of OPDM in these families. And that's because in the few years before we made this identification, groups all through Asia had found that CCG or CGG repeat expansions cause OPDM. So in Japan and in China and in Thailand, CGG or repeats in the UTRs, so the bits of the gene just upstream of where the protein starts, so the little bit of DNA sequence just before the protein starts to be made, cause OPDM. And we had been collaborating with, there's a really well-known group at Queen's Square Neurology at UCL. We've been collaborating with them on repeat expansions in ataxias and in Canvas, if people have heard about Canvas. And so we said to Andrea, well, we think we might have an OPDM expansion. And he came back and said, well, we think we have one too. And we'd identified the same expansion. So we'd identified it in the Australian families, and they had found in 2 UK families out of their cohort of 300 odd myopathy patients that it looked like there were quite large expansions. So there were around 100 and 120 repeats in these 2 individuals. And when they went and looked at what the phenotype or the diagnosis of those 2 patients were, at the time they just thought they just had the umbrella label myopathy. They actually had OPDM, so they had the same muscle disease. So we now know of, we've phenotyped quite well about 35 affected individuals from 8 different families. Philippa identified a 3rd family in her clinic that also has this repeat expansion. And the UK groups sequenced about 50 or 60 small families with OPDM and identified 3 French families with this repeat expansion as well. So the French people aren't going to be very happy when they find out that they're more closely related to British people and Aussies than they might have thought they were previously. We also collaborate with Ira Devison at Sydney, who's an early adopter of long-read sequencing. So when I talked before about next-generation sequencing, I said we take DNA and we make little fragments and we sequence these fragments and then we stitch them all back together, and that's the way we can sort of do the sequencing in a parallel way. There's now these newer technologies where you take really long strands of DNA and you feed it through a nanopore and it determines the sequence. And so using these long strands of DNA, you can actually size things like these repeat expansions. We can't size them if the DNA is all chopped up. So we sent DNA to Ira from the first 2 families, and then we showed this is just how long the repeat is from this technique. And so in some individuals, the repeats are around 100, 120 repeats long, but in this individual it was really long, about 600 repeats. And the UCL group were trying to get it to work over there, and then they decided that Australia was doing it much better than they were in London, so they sent DNA from all of their families to Ira as well. So it was a win for the Aussies, really. And Ira sequenced 19 individuals and showed how long the expansions were in all these individuals. And we don't quite know why, but for some reason, the expansions in affected women seem to be twice as large as in affected men and much more variable, so the range is much larger for women compared to men. We still don't understand why that is. We phenotype the disease, so this is really important. I'm not going to go into all the symptoms, but really important in terms of describing the disease, so that when this paper comes out, other clinicians can recognise the condition. The same with the muscle biopsy. It's really important to describe the muscle biopsy so that it gets recognised overseas and additional diagnoses can be made. And then we tried to do a little bit of work to work out how this repeat might be causing problems in the muscles. So we took patient muscle and we extracted RNA. So we have DNA, and DNAs make protein, and on the way the DNA makes RNA, which is sort of the message that ends up making protein. And we could see, we looked at expression of all the genes in the muscle, and the only gene that was overexpressed was ABCD3. So it looks like this expansion in this gene somehow means that there's more ABCD3 being expressed in muscles from these patients with OPDM than in control muscle. And we know one of the things that happens is that sometimes these repetitive sequences in the RNA, they can aggregate and form sort of toxic foci, or sort of toxic aggregates of RNA and proteins, and then that interferes with function and causes toxicity to the muscle and muscle damage. And so we used a probe to look for this expanded RNA, and we could see, we did this in fibroblasts, so that's sometimes why we might ask for a skin biopsy. Sometimes we get skin biopsies, and then this fibroblast, we can just stick it in a dish, and the cells grow out from the skin biopsy. And then we have cells for a long time, and we can do experiments over and over on these cells to try and work out what's going on. And we can also reprogram them to make iPSCs or pluripotent stem cells. So anyway, we got the skin cultures going, and we could see these sort of, this red signal are these RNA foci in the patient fibroblasts. And we also looked at the muscle biopsy, and we can see again within the nuclei, so the blue staining are the nuclei, we can see sort of aggregates of these mutant RNA within the muscle nuclei, and this is part of the pathology of the condition. So we think that this is one way that this repeat expansion might be causing the muscle disease. And then the very last thing I'm going to say is that I've talked about next generation sequencing. We also have next generation cytogenetics, which is sort of an imaging technique. So instead of sequencing DNA, we take really long strands of DNA, almost the whole chromosome or half a chromosome. That's how long the strands can be. And we label it with a fluorescent dye at a particular sort of sequence of DNA, and then we stick it through on a chip that makes the DNA linearize in these nanochannels, and then we can sequence it with a fluorescent microscope. And this is a really good technique for looking for different types of structural variants. So if we've got changes in the chromosomes, rather than a single typing error, if we've got, half the book's being put at the front end instead of the back end, we can see it with this type of technology really well. And the bio-nano optical genome mapping is one of these approaches, and it has a special package that allows diagnosis of FSHD, and traditionally genetically diagnosing FSHD has been really challenging. So this is the machine, an image. So each of those blue lines is one of these linearized strands of DNA being imaged through the fluorescent microscope, and the little green dots are sort of where the probes are, and then we can take these massive strands of DNA and map them back to the reference genome and try and look for these structural changes in the DNA. And so we got a grant from FSHD Global, and we bought one of these machines, and we installed it at PathWest and employed Dr. Harmony Clayton to optimise this process at PathWest. And Harmony, a few months ago, applied to NATA, those are the people that accredit diagnostic tests, to have bio-nano accredited for the genetic diagnosis of FSHD. So this is the first bio-nano instrument in a diagnostic lab in Australia, and the first time that NATA accreditation body will see an application to accredit optical genome mapping for a test. And this is really important because there's this huge need to properly diagnose FSHD patients in Australia, and as we move to clinical trial readiness, we need a diagnosed population. So this is something that FSHD Global really identified as an area that they wanted to work on. So there have been many novel disease genes to identify and characterise, and there are likely many more to uncover. We now have new reference tools. Recently, the human genome was actually completed. So we talked about the Human Genome Project, but there were a few holes in it. So there's now new reference sets that we can use as well that will help along the way. We have these new long-read sequencing and optical mapping approaches, and all of these, I think, will help us to solve the unsolved. I hope I've shared that deep phenotyping and really good communication between the research team, the clinical team, the diagnostic team, and the patients can be critical to making a genetic diagnosis. And we're now in a new phase of genomics where I think we're beginning to reach for the high-hanging fruit, or as our colleague in Montreal likes to say, the sunny apples. So I hope that in future years we might come back and share some of those sunny apples with you. And just a brief mention that the Australian Neuromuscular Network was formed, it's a national network, was formed many years ago by Nigel Lang and Kathy North, who's since moved on, and other individuals in the field. And then it sort of went into a hibernation, I guess, over COVID, but has recently been reinvigorated by Ian Woodcock in Melbourne. The Congress is every year alternating between the Sunshine Coast and the capital cities, and the plan is that in 2026 it'll come to Perth. And with that, I'd like to thank my team of staff and students at the Harry Perkins Institute and at Pathwest and our funding bodies. And thank you for your attention, and very happy to hear how we could maybe better serve the neuromuscular disease community in WA. Thank you.
Wow. Whose brains are... I'm just going to grab that quickly. Just so you know, it is a real mic, but it's not loud. It's actually just for... This is the... That's okay. It's okay. This is being videoed so we can share it with all of our regional community and for anyone else that wants to see it later. So Piper's going to come round. Piper's going to come round.
I don't know what I've pressed on my phone.
That's okay. Piper's going to come round. If you have questions, just... Right, we'll start right here.
Mind blown is just the tip of the iceberg. I had a question. You mentioned that ataxias and neuropathies are often diagnosed in later life. Why is it so much more difficult, and do environmental factors influence gene mutations as you go through life?
Yeah, so most of the diseases we work on, the mutation is there from before birth, but the disease manifests later in life. In terms of us being able to identify it, often in the paediatric setting, for example, you have a child with a condition, both the parents are well, and then we do a trio analysis where we look for variants present in the child that are either absent from the parent, so it's a de novo dominant mutation, or we look for recessive mutations where where each parent is a carrier, and then we look for those two variants coming together in the affected child. So we can do a bit more filtering in terms of, because we have that family structure. Also, some of the later onset conditions, there might be people in those controlled population databases with the variant, but they just haven't manifested the condition yet. And so the filtering in terms of knowing, well, is that disease causing or not, is difficult. Whereas if you've got a child with that condition, they will know the disease is present before they'd end up in a controlled data set type thing. There's also some of the ataxias and neuropathies that can be reduced penetrance. So not everyone, and like for motor neuron disease, for example, is a good example of that, where not everyone that has the disease-causing mutation will actually develop the disease. And so if you're trying to do linkage and you say, well, this person isn't affected, but actually they're carrying the mutation, they just for some reason aren't going to develop the disease, that can make it really tricky to try and identify the mutation. And the ataxias with those repeat expansions and contractions and the structural variants are more difficult to identify, and so that's part of it as well. So there's lots of different reasons, but they all come together to mean that, yeah, it's more difficult in sort of the adult setting than in pediatrics. And often we want to find out, did your parents have the disease and did they have the mutation? And if you've got a patient in their 60s, well, their parents may have passed. So you're never going to find out or get their DNA to do those sorts of studies. Yeah. Mm.
Hi, thank you so much. It's really informative and interesting. With the identification of the gene and then how it's been changed, have there been any cases where it's been able to be modified or repaired so that the disease has been either cured or...
Yes, so that's the next step, and I think what Rhonda's going to talk about is sort of developing therapies for some of these conditions. We can't develop therapies for all of the diseases that we identify because each of them is many decades of work for each for each gene. In some cases, it might not be that there's a treatment that can modify the mutation and cure the disease, but there are sometimes some examples where you might identify the causative gene and that pathway can actually be treated. So there's some examples where you can supplement the defective pathway, and that can actually improve quality of life quite a lot. So I think they say in rare disease patients about 20% of the time it sort of changes clinical management of the patients, or there might be something that can be done in terms of not a curative therapy, but sort of a treatment that could modify quality of life or function.
Wouldn't it be nice if it was just as simple as being able to just put it back to the way it was?
It sounds easy, but yeah.
Any more questions?
Yeah, how that one change in the 3.2 billion.
Thank you. That was fascinating and mind-blowing. I'm just curious, is there any research into myasthenia gravis or genetic components in that?
Oh, not that we're doing. Yeah, I don't have a good answer for that. I can come back to you.
Okay, thank you.
Have you actually been in touch with, they've got a patient organisation that
Yes.
may know. I don't know if they
I'm
are aware. I could
part
put
of
you in
the-
touch with them.
Myasthenia gravis Australia, like I'm in touch with a lot of, there's one in Queensland, specific. I just feel like I've only recently been diagnosed, but there's a lot of similarities with my dad with similar symptoms. And I was just curious to know whether there's anything, he's 92 now, but I don't know. I've sort of queried it with my neurologist, but I'm very early days and he sort of didn't really say anything about going there. But I feel like if there is something, it's important to know these things. And
Yeah, we can exchange emails.
okay,
Yeah, that sounds
fantastic,
good.
thank you.
I would recommend just getting a DNA sample off him. If he's 92, I'd take a sample just in
Yeah,
case.
okay, yeah, I know. I think
Just
so.
a saliva sample. We can
Yeah.
send a tube.
Yeah, okay, fantastic, thank you. Thank you.
Any more questions? No, I have a couple. Can I just ask, the costing of the sequencing? You said it started out in the billions or the millions. And what is the costs associated to? Is it like the equipment? Is the equipment super expensive? Or is it the time that it takes to analyse it? Or is it a combination?
It's a combination. So when we say $1,000, that's typically sort of the cost to all the DNA handling costs and then generating the sequencing data. That's not any of the analysis time. I suspect the actual analysis time is more expensive than the sequencing, and they're now talking about we're going to be down. There's some other new way of doing sequencing that makes it even cheaper, where they're talking about a $100 genome. And sometimes when we send collection kits away, it costs us like $15 for the Australia Post envelope and $20 for the kit and $15 for the postage to come back, and then I don't know, probably about $20 to do the extraction. So that's almost $100 just to get the sample. So we're talking about, we're getting to a point where almost just getting the sample is going to be the same cost as the sequencing. But a lot of the cost is in the storage of the data. So people forget the data is really, there's a lot of data. It costs a lot of money to store it in all these clouds all over the place. And then, the analyst time as well is also really expensive, But that $1,000 is mostly kind of sequencing costs, and a lot of the sequencing companies, all the reagents are proprietary. You don't know what's in them. You have to buy it from them. So the machines used to be quite expensive, and most of the companies, my understanding, have turned to a model where they give you the machine, but then you have to, all the reagent costs are really expensive. Yeah.
My other question is, the BioNano new modelling or machine or whatever, the process, who figured that out?
Don't ask me.
I mean, that is pretty
Yeah, I haven't looked into
smart,
the
but
history of
is,
that.
like, yeah, I was just like, wow, that's amazing technology. Other question is, you spoke about 10 families that have a condition across the world. Now, what are the chances, or am I just being ignorant, that they are actually related way back when?
No, that's a really valid question. So there are examples of that. So they would have to have the same mutation. So these families all have different mutations, so they're not related. But there are some examples where they probably do all share a common ancestor. So for example, one of the diseases we identified for genes we identified for nemolymopathy, it's present, that variant is present in 1 in 500 Japanese people. It's a recessive condition, so you have to inherit 2 mutations. But 1 in 500 people in Japan carries that mutation. And one of the other mutations we found, I think 1 in 250 Chinese people, they think carry that mutation. And they showed it arose 6,000 years ago in southern China, and that basically everyone carrying that mutation comes from a common descendant. Yeah, so it does happen. And we think potentially that the Australian family here and the Australian family in Melbourne are probably related.
Okay. And then I was just also going to ask, for anyone that's more interested in research, I think you've demonstrated the importance of your multidisciplinary teams and linking with clinicians and researchers, and obviously the patient is part of that process. What more can be done to create those linkages? And I guess what role can patient community members and play in that?
Yeah, so we have a consumer reference group that we work with that we probably need to refresh a little bit and could get new representation on that group. Yeah, I don't know how. Yeah.
It was just a thought.
Yeah, yeah, good thought.
Captive audience.
Yes, yes, and if there's things that the community, would like from us, let us know. If people want to come and visit, or I was just thinking, for Rare Disease Day next year, maybe we should start thinking about doing some sort of day around Rare Disease Day at the Institute with a neuromuscular focus. Yeah, very happy to explore options, and people are always welcome to email us as well.
Awesome. Oh, we have one more.
Just bear in mind, I'm not a clinician, so I can't give health advice things, but, happy to talk about genetics. Yeah.
Thank you so much for the presentation. Very interesting. I do have a genetics degree, so, and then to get diagnosed with a rare disease, it's really interesting how you've got that multidisciplinary network working, but at least from my experience, the patient actually isn't even aware of that or isn't even part of it. So there isn't any communication with the patient from my perspective at all. I'm wondering how I can get some of the research papers that you're publishing so I can read them and inform myself, because ultimately I'm responsible for my journey through the disease. The Neuromuscular WA has been wonderful with their help, but there seems really exciting stuff going on in the genetics and the research, and you've got your multidisciplinary team, but there seems to be a lack of information coming back to the patient. What's happening in that sphere? Am I wrong, or was I just visiting my neurologist on the wrong day or something?
I don't know, it's hard to say. And because we, in the research lab, typically we don't interact directly with patients. We do occasionally, because sometimes they find, patients will find us because they'll Google myopathy and find me, and, or Rhonda or Nigel, and then they email. So we do. And it's something also, I think, we've sort of been a little bit hesitant to do it because we're not clinicians, but I think the world is changing. So I think in the old days it was very, there's distance, but I think things are changing, and I think I'm probably up for the change. Like, I might get things wrong occasionally, or not know, and I'll come back to you, because that's not my area and I don't want to get it wrong and I don't know about that set of conditions. But yeah, I think if we're open to it and the patient community is open to it, that we should be able to interact more. Yeah.
So when you work that out, I'd like to know.
Yeah, I mean, I don't think we could have patients at the MDT meetings, for example, because we're discussing lots of patients and it's all still preliminary. There's variants we're not sure of and there's confidentiality things. But yeah, how we might engage a bit more broadly, yeah, we can think about it, yeah.
And if you're able to give us access to some of the papers you've published.
If there's papers you're interested in, let us know. We can send them.
Okay, thank
Yeah,
you.
yeah.
Awesome. Oh, we have another one.
Hi, I'm a FSHD late onset patient. I just want to ask about the clinical trials that are for the eastern states just recently, but they're not going to be happening in Perth at all for the FSHD. Is there a process as to, you know, when you get the go-ahead for here, but the eastern states do, but we don't here?
Yeah, it's tricky. I think the eastern states perhaps have been more experienced taking trials on or have done more trials, and so they use, you know, and I think it takes a little bit of money. My understanding from talking to some of the neurologists is that there's a bit of a outlay in terms of money to get the trial set up. But then once the trial's running, it sort of pays for itself and it pays for the next trial because they're quite lucrative. But I think perhaps, and I had some grants in with Royal Perth that weren't successful to the state government to try and improve or upgrade and get some extra hands into the neurogenetics service and part of it was to do the registries and get clinical trial ready for some of these conditions, and the funding to the state government didn't come through. From state government didn't come through.
Yeah, I
maybe
liaise a
that's
little
something.
bit with Emma.
Pardon?
I do liaise a little bit with Emma.
Yeah.
And yeah, it was just a bit disappointing that the 4 sites are going to be set up over there, but nothing here.
Yeah.
Which I said I'm prepared to go to Melbourne or wherever that I need to.
Yeah.
Yeah, just feel a bit like Perth gets left behind sometimes.
I know, and it's hard because I'm a researcher. There's no way that I couldn't do the trial, but yeah, trying to work with our clinical colleagues to see how we can facilitate it, and with FSHD Global, or you know, I did say to Catherine, I think I put Catherine in contact with Ian and said, you know, talk to him about getting your FSHD clinic set up. And
Yeah. Well, I've got 6 of my family here, you know.
Yeah.
It is such a tricky thing because
it's hard,
it's not unique to FSHD, it's across all conditions.
Yeah, that's right.
But it's really, as Gina said, it comes down to the infrastructure that's in place in Perth to facilitate clinical trials. And the actual costs to do that, it's not just running the trial, it's having the clinical trial coordinators, it's having the physios to do the assessments, it's having the access to the databases, and so the costs add on, and then you multiply it with all different conditions. And I guess you've also got to get the site approved by the farmer. But look, any questions like that as well, if you're looking to advocate or if you have questions on that, reach out to us. We probably won't be able to influence it, but the more we have these conversations and the more times we can put it in front of the health minister that more clinical trial readiness is critical in WA, you know, that's the role we can play, is we can advocate for it, and it won't be for a specific condition, but it will be across the board to improve, I guess, the health system bit to be able to facilitate that.
Yeah. sorry, don't have better answers than that. But we have tried. We have tried, and if you could advocate to health ministers and things to try and, because I think my understanding, and it may be very naive, and I may have to apologise for this, but my understanding is that the initial outlay is not that much money or time to get to a point, and then you're at a point where actually pharma's quite lucrative, and it'll it'll fund itself. And once you've got one trial, it's easier to get another trial. But I can't run them, and I don't run them. But that's my understanding of those initial kind of outlay issues.
Okay, and on that note, I think we should have a break. So everybody can have a toilet break. Gina, thank you so much.
Oh, you're welcome.
Amazing. Very much appreciated. And we'll have you back. So please help yourself to some food, grab something to drink, go to the loo, and we'll be back in a little while. Okie dokie. Take two. I'm going to have to read Rhonda's bio intro. Not that couldn't remember Gina's, and won't be able to remember Rhonda's. But so our next presenter is Dr. Rhonda Taylor, and Rhonda's presenting on working towards developing treatments for muscle disease. Dr. Taylor completed her PhD in 2015 at UWA. She now leads the disease models and therapies team with Preventative Genetics Group at the Harry Perkins Institute of Medical Research. Rhonda's research aims to develop novel genomic treatments for congenital myopathies. Specifically, she leads projects that involve development of therapeutic strategies involving CRISPR-Cas9 gene editing, oligonucleotides, and RNA-based therapeutics. Rhonda is also developing and characterizing human cell models of congenital myopathies, which she hopes will facilitate the development and testing of novel treatment strategies. Rhonda enjoys engaging with patients and consumers, is passionate about gender equality, is a UWA ally, and is a supporter of early to mid-year career researchers through EMCRs. I really should know that. I'm reading this acronym. Early Mid-Year Career Researchers. So welcome, Rhonda, and thank you very much.
No worries. Thank you so much for the opportunity to come and connect with you and speak with you today. As you've heard, I work quite closely with Gina through my connection to Gina and Nigel, but I don't work in the diagnostic space, and so my role is actually to try and develop treatments. This evening I'm going to be telling you a little bit about the development of treatments in general, although this is a really, really, really broad space, and by no means what I'm going to tell you is comprehensive. So I'm just going to be giving you a little sprinkling of a little couple of examples about some treatments that have been developed and how they've been developed, before I lead into what I'm trying to do in my work here in WA. So I'll start with just telling you a little bit about me and how I got here. So like Gina, I completed my undergraduate degree at UWA, and that's where I really fell in love with genetics and kind of became a big DNA nerd. And I followed my undergraduate degree with an honours in genetics, and then also I started my PhD in 2012. So the focus of my PhD was understanding a really, really, really small component of what we call genetic risk for an autoimmune condition called lupus. And lupus is a complex condition. So it's in the name. It's got a lot of very different genetic factors and a lot of different environmental factors that all interact in this really complex way. And it's a really difficult problem that people don't really understand yet about how to untangle that in order to lead to prediction of who's going to get any given complex disease, or also how do we treat them. So when I came to the end of my PhD, 2014, 2015, I felt that what I was doing was really important work, but a long way from reaching a patient. And I felt that I wanted to make more of an impact sooner. I wanted to be working more closely in an area that I felt might be able to reach people one day and have more of a positive impact on people's lives, or more of a direct positive impact on people's lives. But I didn't really know what I wanted to do, so I looked around in Perth for genetic research that was being done with more of a translational focus, so more of a direct impact on people. And that's when I learned about the work being done by Nigel Lane, Kristen Novak and Gina Ravenscroft at the Harry Perkins Institute. So led by Nigel and Gina, they were really looking at the genetic causes of neuromuscular diseases, as you've heard. And at the same time, Kristen was leading work to try and develop treatments into some of the conditions that they were diagnosing. And I kind of immediately knew that that's what I wanted to do. I wanted to try and develop treatments if I could. And so I joined the team. And a couple of years after I joined, Kristen left to become the director of the Office of Population Health and Genomics at the WA Health Department. And then a couple of years after that, I kind of took on Kristen's role as the leader in that space in the team across the groups of both Nigel and Gina. And that was in 2015 that I first joined the team, and I'm still here. They can't get rid of me. I really love my work. I love what I do. But before I tell you about what I do, I'm going to just go right back to the start and give you all a little bit of my undergraduate degree. I'm going to give you a 101 in genetics, just so that we're all on the same page with some of the terminology that I'm going to use, because it's going to be important to understand later what I'm trying to do. So here we have a cell. There's lots of different types of cells in your body. This one might be, for example, a skin cell. Inside every cell, there's a little compartment, a little pocket called the nucleus. And inside the nucleus, humans have 23 pairs of chromosomes. Each chromosome, they often look like this. They're often represented as these little X's. Chromosomes are really just tightly wound up DNA. Like Gina said, it would take 27 years of typing, so the DNA itself is very, very long. But to get it inside the cell, it's packaged up really tightly into these chromosomes. And importantly, each chromosome comes as a pair, one of which is inherited from your mum and one of which is inherited from your dad. If we look more closely at the DNA, we can see these look like these two long strings that are wrapped around each other, and we call these two strings together a strand, or a single strand. And I've put boxes here along there to illustrate that the strand is long, and along that strand at various points there are specific sequences in the DNA which are your genes. And a gene is basically just a discrete section of the DNA that encodes for a functional product. And a lot of the time that product is a protein, which is what Gina talked about. But to get between the DNA and the protein, you have to go through an intermediate molecule which is called RNA. So basically what happens is the DNA acts as a template from which you make the RNA, and you can see the RNA is just one strand, whereas the DNA was two. And then from the RNA, that provides the instructions for making the protein. And the protein is made up of individual units called the amino acids, which are then stitched together in a string. And it's the different amino acids in the string, their order, their composition, their length, they're all combined together to give a protein its function. And there's lots of different types of protein in the body. Proteins are basically the workhorses of the cell. So they can be structural, they can give the cell its shape, or they can also catalyse basically all of the reactions that are occurring in your body at any one time. And humans make about 20,000 different proteins. So there's a lot of different, and each of those, not necessarily one-to-one, but mostly each of those is coded for by one gene. So some proteins that you might have heard of, keratin is a common protein in hair, collagen is a common protein in skin and bones, and albumin is a common protein in blood. So that's just to kind of give you an idea of these are examples of proteins. Now, if you haven't followed a lot of that slide, that's okay. The key things I really want you to take away from that information is that DNA contains genes which code for your proteins, and in between the DNA and the protein there's this intermediate molecule called the RNA. So that's important, the flow of information is important there. If you make a change to the DNA, that change gets carried through into the RNA, and then that will carry through again to a change in the protein. And so another important take home is this point that I said about there being two copies of each chromosome. That also means there's two copies of each gene. And an important concept in genetics is dominant and recessive, which are terms you might have heard your clinician say. Basically, that relates to whether one or both of the copies of the DNA or the gene contain the change. So dominant is where only one of the gene copies needs to contain the change in order for the disease to happen, and recessive is where both of the gene copies have to contain the change in order for the disease to happen. There's another category called X-linked, and that's when the damaging DNA changes on the X chromosome, and that will affect males and females differently because females have 2 X chromosomes and males only have 1. The reason I tell you all of that is because when we come to developing treatments, whether the disease is dominant or recessive makes a difference, because dominant and recessive diseases cause the change has a different effect, and we have to understand what the effect the change is having on the protein and on the cell in order to be able to actually design the treatment that's going to target that cause or the mechanism. So, what are some examples of some mechanisms? How does it actually happen? How do you get from this change in the DNA to a muscle disease? So recessive disease, as I said, is where you have one copy from mum and one copy from dad of a gene, and they both contain a DNA change. And I've shown the DNA change as the little star. That's still present in the RNA. And in recessive diseases, often what happens is this change is actually leading to a loss of the protein. So it's causing perhaps the RNA to not make a protein at all, or it's causing a protein that is made, but it's just completely lost its function. So we call this a loss of function mechanism. And if you think about, okay, you just don't have the protein, how could we treat that? You can treat that by perhaps just giving more of the functional protein or more of the functional gene that can then make the protein. And so an example of that is called gene replacement therapy or just gene therapy. Zolgensma is an approved treatment for spinal muscular atrophy in babies, and this is essentially a gene replacement therapy. The patients are given a functional copy of the gene that can then make the protein that's absent in the babies. And so that's how that works. X-linked variants are sometimes can be treated similarly as a loss of function. So a good example of a loss of function condition that's X-linked is Duchenne muscular dystrophy. And we're really fortunate in WA that we have a lot of expertise in this space because some of the approved treatments for Duchenne muscular dystrophy were actually developed in WA. So one of those types of treatments is called exon skipping. And basically the Duchenne muscular dystrophy is caused by loss of function variants in the dystrophin protein. So this is dystrophin here, this big long sort of greeny blue molecule. It's a really, really, really big protein, and its function is to essentially hold different parts of the muscle cell together. So it's got to have these 2 ends intact that can grab onto the 2 different parts of the cell, hold them together, and that basically gives the muscle cell structure. What we see in DMD is that part of one end of the protein is kind of missing, and therefore it can't do its job. It can't link those 2 bits together because it doesn't have this sort of linking part at one end. And so here at Murdoch University, Sue Fletcher and Steve Wilton actually developed these drugs that are in clinical use for some types of Duchenne muscular dystrophy. And they're pictured here with Billy, who was a participant in one of the original clinical trials for these exon skipping drugs, and these are the 3 approved drugs. And as I understand it, Billy is kind of in university now and he's still walking, which is wonderful. But how do they work? What do they do? Well, if we say that there might be a variant here in the dystrophin protein, what the exon skipping drugs do is they actually cut out the middle part of the protein that contains the change, and that leads to a shorter dystrophin protein that still has both its ends, and therefore it can still do that job, or it can do the job maybe not quite as well as the full-length protein, but it's giving the cell more structural integrity because it's got those 2 ends and it's linking the 2 parts together. So a kind of key take-home message from that example is that the exon skipping strategy was able to be developed because we understood what was happening in the muscle cell as a result of the specific genetic change. So understanding the genetics and understanding what the effect of the change at the DNA and the protein level is enables us to kind of make rational design choices about how we might treat that disease. The exon skipping strategy works in DMD because the dystrophin protein is really large, and it can skip part of the middle of the protein, but still give the protein some function. But this won't necessarily work for all kinds of proteins or all types of diseases where the protein might be shorter or it might just have a different structure, and so we can't kind of apply this as a one-size-fits-all approach to treat every type of disease. In addition, the drugs only work for specific genetic changes because they act at that RNA level, which is very sequence specific. You can only target, one drug can only bind to one part of the RNA and lead to one type of chopping out. And it took 20 years from seeing the first promising result in the laboratory to getting that drug to use in humans. And there's a lot of different steps between having that idea about how this might work to actually having that work all the way through and be approved for use in humans. But because we now have this pipeline in place, more drugs are being developed that work in this same way specifically for Duchenne, and they're not taking 20 years every time now. So we're kind of learning from the examples that we have, and hopefully this also paves the way for similar types of drugs to be used for other conditions that are not Duchenne. Okay, so that was kind of an example of a recessive disease, or in this case an X-linked disease, and how we might treat it if it's a loss of function. On the other hand, we also can have dominant variants, and they can cause disease in a different way that's not necessarily a loss of function. So one way that dominant variants, where you've only got one copy of the gene is got the change in it, but one copy is the healthy copy. One way that dominant variants can lead to disease is what we call haploinsufficiency, and it basically just means that you don't make enough protein, you need more protein. You need both copies to be making protein to have enough for the cell to function well. And if that's the case, you can kind of treat that in a similar way as a loss of function. You could perhaps just deliver more, and that would be one way you could treat that type of disease. But actually, the haploinsufficiency is not the most common way that dominant variants cause disease. Often what we see in dominant variants is what we call a gain of function. And that means that the protein is made from the copy that contains the variant, but the protein has an additional function that it doesn't normally have, or a toxic function. And that means that the protein that contains the variant can actually go in and stop the healthy copy from functioning. So it's what we call a gain of function. It's preventing the normal function of the one healthy copy that you still have. And so in gain of function types of diseases, then one way that we might treat that would be to try and get rid of the dominant or the toxic protein. Instead of delivering more, you might just try and get rid of the toxic copy, and then that would hopefully restore the function that you have still on the protein that's being made from the normal gene copy. And there's some good examples of this too. So the example I'm going to give is for FSHD. The trial I'm going to mention now is happening in the United States. It's currently still recruiting, so that means it's kind of in some of the earlier phases. One of the genetic causes of FSHD is gain of function variants of the DUX4 protein. And Avidity Bioscience are the company that are running this, have developed this drug and are running this trial. And basically what they're doing is they've invented a strategy to knock down the DUX4 protein by targeting the RNA. So remember that the DNA goes to the RNA, which makes the protein. So they're not changing the DNA, but they're sending in what's called a short interfering RNA, which is kind of, if you can think of it like a little molecular pair of scissors. And what this is doing is it's going in and causing the RNA to be chopped up, which means that it can't be used as the template to make the protein. So if you get rid of the RNA for this DUX4 gene, then you reduce the amount of protein that's made. I will note that in this example, the treatment is targeting both the copy that contains the variant and the healthy copy as well. But their preliminary results in mouse models showed that this still had a therapeutic benefit. So there was obviously enough protein still made from the normal copy that could still result in improved muscle function. And so they treated some mice with this drug that they've called AOC 1020, and they saw increased treadmill running and also muscle force measurement. So that's indicative of an improvement in the muscle function. Like I said, they're now recruiting for a phase 1-2 clinical trial. I think they started recruiting in 2022. So we might start seeing some results coming through from that soon. And I think that will be really interesting to kind of keep an eye on in terms of how it's working in patients. They also have a similar trial for DMD. And if you want a bit more information, you can kind of, even just Googling Avidity Bioscience, FSHD or DMD will bring you up lots of information. Okay, so I've given a couple of examples about how you might go about kind of thinking about how do we do a treatment for a disease. And the examples that I've given kind of give us a little bit of a pipeline, like how could you go about it in more of a general way if you wanted to try and develop a treatment for a disease that didn't have a treatment. Well, the first step was understanding the genetic cause of disease, and then understanding how that genetic change leads to the features of the disease that we see. How does that change at the DNA? What proteins does it impact? And how does that lead to a loss of muscle function? If you can understand those things, then you might be able to think about how would we tackle that. For example, if it's a loss of function or a gain of function, what tools do we have that we could use to try and overcome that? And then the next step would be to obtain a cell or an animal model that replicates the features of the disease that we see in humans, in the dish or in the animal. And that is a really, really, really key step of treatment development, because we have to be able to show that a drug can improve those features. If the feature is muscle weakness, we have to be able to show, before we go anywhere near a human, we have to be able to show that the drug we've developed can improve that feature, can improve the muscle performance. And both the examples that I gave for both DMD and for FSHD had really good animal models, which meant that we were able to actually test, do those treatments work? So it's a really key step to be able to have a model that replicates the features of the disease in a way that we can test it in the laboratory. And then after that, if you show good feasibility of your idea and your drug in a cell and an animal model, then you'd want to start moving towards assessing safety and delivery and thinking about clinical trials. But this first step of the preclinical development we have a lot of research in this space, but it's a really big bottleneck, getting things from the preclinical stage through to reaching patients. And in my opinion, one of the reasons for that is not having good enough cell and animal models. Okay, so here's a little schematic, apologies it is a little bit small, of kind of some of the things I've touched on there. What is the pipeline? And it's, you've got to kind of have an idea, design it, study it in cells, figure out how it works, and then test it in this preclinical models that I talked about, cells and animals. That's basically T0 of the translational pipeline, so step 1. And then after that, you start moving through to your different phases of clinical trial. So if we have a pipeline, we kind of have a bit of a roadmap. Why aren't there more treatments? And this is a really big question, and it's got so many moving parts to it that I can't possibly touch on all of the reasons and all of the hurdles that there aren't more treatments. But I will just touch on a couple of them. So we know that rare diseases as a whole are not actually rare, but unfortunately 95% of rare diseases still lack an FDA-approved treatment. So this is a huge unmet need. We know we need more treatments. But some of the reasons why it's, again, this is just my perspective, but some of the contributing factors are that because each condition is rare, there's a very small number of patients potentially with each condition. Historically, that has led to a lack of investment from pharmaceutical companies that don't see a huge dollar sign with only maybe 10 patients worldwide potentially wanting to buy the drug. Also, many conditions are lacking, again, probably partly because of their rarity, they're lacking natural history information, which is something you actually need to be able to move into a clinical trial. So we don't have that information. Even if we have a drug that looks promising at the preclinical stage, you can't really move it through to a clinical trial. There's also, as was touched on earlier tonight, a lack of patient involvement in all stages of drug development, and even at diagnosis. But basically this means that I might come up with an idea, but not having spoken to many patients or not having much of an understanding of the patient experience, I might design the treatment and it doesn't target the right thing because I haven't maybe spent enough time speaking with patients. And I think this is a common thing that's happened historically, that maybe the drugs are designed are not desirable actually to the patients, or they don't target the right outcome measures. Okay, and then the last thing is that, again, it's not the last thing, but it's the last thing I'm going to talk about, is the high cost of drug development. So even at the preclinical stages, it is incredibly hard at the basic research level to secure enough consecutive funding to actually make good progress. In addition, I touched on the absence of those cell and animal models. They take a long time to develop. You know, as I'll show you in a minute, I've been working to develop some new models for 5 years now, and that's how long it's taken me to get the model to even a certain point that I think is reasonably good. And so that's 5 years of funding I have to find for myself and my staff, all of the reagents, all of the things. It's a long time, and it's not even at the treatment yet. So it's really hard, I think, to just kind of make enough progress with insecure funding. So I'm now going to switch gears and tell you a little bit about what I have been able to achieve in that time, be a bit more positive. And so the example case that I'm going to talk about is nemaline myopathy, because as Gina mentioned, it's one of the ones that, you know, was discovered by our team and that our team has worked on for a lot of years. So we have a bit of a background in this space. So nemaline myopathy is usually discovered in babies at or shortly after birth. There is a range of severities with this condition, but unfortunately many babies do die within the first few years of life and there's no cure. And in terms of the genetics of nemaline myopathy, it is caused by a range of genes, as Gina said, but one of the first ones that our team discovered is called skeletal muscle alpha-actin, or ACTA1, and so that's been the focus of a lot of my research, is this specific gene. And we know that 10% of the genetic variants in ACTA1 cause recessive disease, and the mechanism there is loss of function. The patients have either a complete loss of the actual protein itself or the function of that protein, but 90% are caused by dominant variants, and those have a toxic gain of function mechanism. So that's where the toxic copy interferes with the remaining function of the wild type copy. So the strategy that we've taken in trying to develop treatments for this, I have looked at trying to develop different treatments for both dominant and recessive disease that target that underlying mechanism. So they're different types of treatments in both instances. But the one that I've spent probably a bit more time and have made a bit more progress on is the dominant ones. So I'm not going to reinvent the wheel. I'm really going to try and stand on the shoulders of giants, because we know that there are ways we can knock down or get rid of the RNA so that the toxic protein is not made. In the case of ACTA1, we don't want to get rid of the healthy copy. You actually need a lot of ACTA1 protein in your muscle cell for it to function, so we can't get away with only having 10% of that normal ACTA1 being there. So we've designed, the point of difference between our study and the kind of what I mentioned before is that what we've done is try and design the treatment in such a way that it only removes the toxic protein and not the healthy protein. Sorry, my laser's off. Only removes the toxic protein but doesn't touch the normal protein. And I've been really drawing on the expertise of Sue Fletcher, who was one of the investigators who developed those drugs for Duchenne muscular dystrophy, in developing my approach here. And I got to a certain point in the design and discovered that I couldn't move many of my treatments forward because I didn't have good enough cell or animal models in which to test. I could do the design, I could come up with an idea, but I didn't have enough of a platform to actually progress the drugs forward where they might one day be able to reach patients. And so I had to kind of sidestep for about 5 years and try and come up with a way of developing better models that we could use for testing. And so our approach has been to take patient blood or skin cells, because those are relatively easy to obtain and the procedures for taking them are relatively not invasive. But the trouble with that is that if you're trying to develop a treatment for a muscle disease, you can't test the treatment in a blood cell. The blood cell and the muscle cell don't have the same proteins, and ACTA1 specifically is not present in blood cells. So we have to test the treatment in the right type of cell. And so what we've done is take those blood and skin cells and give them the instructions for turning into a different type of cell, which is called an induced pluripotent stem cell. And stem cells are special cells. Stem cells can turn into every cell in your body. So what we've done is take the patient blood, turn it into this stem cell, and then tell the stem cell to turn into a muscle cell. So now what we have is this collection of patient-derived muscle cells that we've obtained from patient blood. I've made that sound simple, but it took a long time to get right. But what's really cool about this approach is A, the fact that it's minimally invasive to take, but when you get to the stage where they're a stem cell, you can make lots and lots and lots and lots and lots of those stem cells. So we now have basically an unlimited supply of these patient stem cells that we can use to make muscle cells, or depending on the disease, we could turn them into nerve cells, or other types of cells too, depending on cardiac cells, heart cells, depending on what you wanted to study in the disease. You can take patient blood and ultimately turn it into these different representatives of different parts of the body. And I still kind of think, to me, that even sounds a little bit science fiction that we can do this now, but we can. And so here is a little example of what that actually looks like. I will say at this point that a lot of this work, getting those protocols right, like actually doing it in the lab, was led by my colleague Joshua Clayton here, who's pictured. And this is kind of what the cells look like, just in case you're interested. So here's an example of kind of the blood cells. This is what they look like when we're growing them as stem cells, and you can see that they look quite different. Here this is an early muscle cell that we've made, and here we've instructed the early muscle cell to form a more advanced muscle cell, which is called a myotube. And it's these long, skinny structures that are a little bit more like the muscle fibers you might have in your body. They're not perfect. They're certainly not the same as what you have in your body, but they're kind of an early precursor, I guess, to the types of muscle that you have in your body. So they're a good system for having an early look at something, at a drug, perhaps, but they're not a perfect model. And like I say, this process took us about 5 years, but we now have at least 4, possibly 5 now, ACTA-1 patients. We've got these cells growing in the lab that we can make nice myotubes from. Since, a bit like the exon skipping example, the first one took a long time, the other ones are a little bit quicker. So we took us a long time on the actins, but we're also now starting to recruit and make cells from other groups of patients as well, which positions us to start developing treatments for some of these other conditions as well. So we started with actin. We've also got 5 different stem cell batches from 5 different RYR1 related myopathy patients and 3 different Lang distal myopathy patients. So we're trying to develop a little bit of a bank that we've kind of got some cells that are representative of different conditions that we can then try and develop treatments for, hopefully, moving forward. And now we're at a really good point where we can actually start testing some of those treatments that I designed years ago but got stuck on because I didn't have the right cells. And so the last year I've spent really trying to get those methods going. Like, one of the first things we have to do is actually get the drug into the cell. And figure out, how do we measure it? How do we say whether it's working? What are all of those sort of methods that we have to do? So I've been working to get those up and running over the past year. And we're really at a good point now where I'm starting to generate some data. I'm not going to show any of that data yet because it's still too early, but I feel really excited about where we are now that we actually have these cells and we have the designs that we can start sort of hopefully getting some preliminary data to say that we might be able to do something about some of these conditions that we have cells for. They do still have limitations, as I touched on. That's kind of the first step, is to say, is it possible? But like I said, the cells that we grow in the dish are still not really close enough to a muscle that's in your body. And the main reason for that is because the muscles in your body actually have mechanical stimulation, they have stretch, they can contract. And one of the main features, or almost a unifying feature across many different muscle diseases, is that they cause the muscle to be weak. So what we really need to do to be able to say whether a treatment is going to work is to be able to say, can it make the muscle contract better? Can we make the muscle stronger if we give it this treatment? And for that, we need to be able to measure this that I'm showing you here, which looks a little bit freaky. This is a muscle cells contracting in a dish. We need to be able to measure that strength. And so what we are doing at the moment is collaborating with a group at the University of Washington who are making these 3D muscle models. They call them engineered muscle tissue. So they take the cells that we've made and they grow them in this system around these 2 posts, and that stimulates them with like electrical stimulation, so sort of like a nerve stimulating a muscle. and then that causes them to contract, and then we can measure the contractile force. So this is really cool. It's a really exciting thing we kind of only just now figuring out how to do. And I think it's going to give us a lot of power to say, do our treatments work? Do they improve this muscle strength? We haven't tested any of the treatments that I'm working on now because we're still at this stage where we're testing them in the 2D to kind of say, what are the lead candidates? Are there any of these that do the thing we want it to do? And then hopefully we'll be able to take those through into this 3D system to get more proof that they actually could have a benefit in a muscle cell. But I'm really excited for what's ahead in this space. And I think it's only going to grow. The more people we can kind of get working in this space and solving problems, because there are still a lot of technical problems. We can make it happen. You can see we can make it happen, but there's still a lot of technical problems around how do you compare one patient's cells to another patient's cells. You know, are they different because they're impaired, or are they different for some technical reason? So there's still a bit to work out in terms of how we actually conduct the studies. But I think we're at a really kind of exciting time. And my hope is that we will be able to take these new technologies and drive forward some treatments for some rare conditions that currently don't have any treatments at all. So a question I often get asked when I talk about developing treatments is, what's the timeline? And unfortunately, I can't give you a definitive answer on that because we're basically still too early in that T0 phase of the pipeline. And a lot of things depend on what do the results look like when we get through those 2D and then also those 3D muscle models that I showed you. We don't know yet whether we have a drug that's going to work. But in order to kind of try and facilitate moving things forward when we get there, because I'm hopeful we will get there, I'm still trying to establish relationships with both consumers. I'm trying my best to reach out and speak to patients who will have the genetic mutations that I'm working on, speak with them, make sure that the treatments might address their needs, and also trying to make more connections to industry partners, because my expertise is working with those cells in the laboratory. I don't have expertise in phase 1 or 2 clinical trials. I don't have expertise in taking things, and interacting with the pharmaceutical industry. I'm trying to learn to sort of know what I don't know so that I can be better prepared to take things there if and when we get there. But those things take time. So previous treatments, like I say, have taken 15 or 20 years to get approved, but we now have those precedents to work on, and we do have current clinical trials that are happening in this space for similar types of drugs that we're gonna learn a lot from those too, and hopefully, with that, there's also changes in the regulatory space that, again, I'm not really across that because I spend much more of my time trying to figure out how do I make this drug go into this cell? What does that do? But there's a continually shifting sort of regulatory landscape as well. And so there are more specific guidelines that are coming out, again, all the time for rare diseases and ultra rare diseases, which I think ultimately will hopefully benefit this community. So we have more resources and knowledge than we did before, but we are still a long way away from that goal of treating more rare diseases. But yes, I have a lot of excitement and hope for what is to come in the next 5 or 10 years. So just in conclusion, there is an urgent need to develop treatments for rare neuromuscular diseases, as we know, because most of these diseases don't have approved treatments. And there's a lot of really promising preclinical and even clinical work happening in the neuromuscular disease space. However, research progress is a lot slower than we'd like, which I know is a huge source of frustration for many. And part of this is related to the high cost and the need for constant development of new and better models for disease, which, because each disease is different, means that one model doesn't necessarily apply to every disease. There's so many variable models that need to be developed, as well as many, many other factors. But to keep things moving in the right direction, I think we really need to be facilitating more and better communication with communities and researchers and clinicians. So I'd just like to finish by thanking the team. A lot of the work was done by Joshua Clayton and various students that I've had in the group throughout the time, as well as research assistants. Christina Vo particularly did a lot of the cell work that I've showed, as well as patients and consumers that have been involved in and supported the work. And we have had a lot of funding from various sources as well that's enabled the work to progress. So thank you. I'd be very happy to take any questions. Yep.
Any questions? Cara.
Amazing. Thank you so much for sharing your research and the work that you're doing. It's just amazing. I have so many questions, but the one I really want to ask you is about the future of, I guess, research innovation in Western Australia as we transition away from a mining kind of state. And I think this space that you're in medical research and then hopefully clinical trials, I really pray that that's the future for WA, because I reflect on the 3 treatments that have been approved for Duchenne. They are FDA approved, they're available in multiple countries, and yet not here in Australia where the technology was discovered. So I think in the treatments that you're looking for, are you mindful of like trying to keep that expertise and that knowledge and development in WA, particularly with some of the NRA manufacturing facilities that have recently got grants? How do you feel about that?
Yeah, I don't have a really clear answer for that, I'm afraid. I mean, I'm mindful of definitely trying to make sure that as many patients as possible can benefit. And I think that's my ultimate goal. Yes. That when I design treatments, I want to make sure that they're going to be able to reach the people who need them. You know, I certainly don't want to design a treatment and then it costs millions and millions of dollars and it's not accessible to anybody, which is one of the problems that, you know, we have with some of the approved treatments. I haven't figured out yet what my role as a researcher is or how I can make a change at that end, because I haven't gone through it yet, if that makes sense. So certainly I'm mindful of it. I don't yet know at what stage, if I was to develop a treatment, how I would make sure. I mean, I think it's tough because there's so many laws and things that, you know, you have to go through often to get the funding, yeah. So the kind of model that we have now is my IP is owned by the University of Western Australia. Previously, I developed a treatment that I was able to show worked in a mouse model, and I took that to the sort of the intellectual property department of UWA, and then they took it and approached a bunch of pharmaceutical sort of companies and tried to get interest, and we had a bunch of, you know, conversations with these different pharmaceutical companies, and we couldn't get any traction on it. And so the reasons for that, they said it was too rare and they said it was too mild, the particular condition that I developed a treatment for. But I get the impression there's like a lot of legalities that would happen even at that first early stage, which would specify who owns what and who has control of what. So yeah, I think you're right, I just need to be mindful of it when I go into those sorts of negotiations, but we haven't gotten there yet to know even what's after that.
My next question, if you don't mind, is sort of at the other end where we are looking at access to treatments. You know, those 3 treatments, the pharma haven't even applied for TGA approval yet for those drugs in Australia. So I'm really happy to see and so pleased that researchers are starting to involve consumers and patients in the design. Hopefully that when a treatment does go for TGA and then reimbursement decisions and navigating that regulatory process, that you can demonstrate that you've got a lot of patient involvement, that it's really treating what the patients believe, and hopefully that will get you approvals and reimbursement, which is the biggest barrier we have for access and very, very expensive drugs.
Yeah, and it's really great, to hear your perspectives and your thoughts and engage with all of you more, because it makes me more mindful of what do I need to consider, which steps and that sort of thing. So yeah, it's really, really helpful. I think this more communication is just going to be benefit for everybody.
Thank you so much.
Yeah.
Any other? Oh yeah, we've got a couple.
Good evening. I'd just like to say that when I came tonight, I didn't really know what this was all about because I came with my wife. But just listening to both of you presenters today, I can't believe how much you guys have to consider. All of the things that must be on your mind. It's hard to imagine that singular people can have all that on you. just want to say it's very impressive, very impressive. And I wish you all the best. That's all I can say. Yeah, both of you.
Thank you very much. Yeah.
Yeah, thanks for the information. I think the graphics really helped as well. I understood you mentioned something about Zolgensma, which is a gene therapy for SMA patients, but I understand that's only for babies. I'm wondering if there is a research or clinical trials for SMA patients for someone older than babies.
I might need to come back to you on that one so that I don't give you any incorrect information, but I certainly can find out for you. Yeah, you're correct that the Zolgensma is just for babies, I think less than 9 months, but yeah, I'm happy to give you my email if you'd like. I can look it up for you. Yeah.
Thank you so much, Rhonda. It was a really interesting, eye-opening presentation. Very conscious of one of the main blocks to progressing forward to real-life outcomes for patients is this issue of funding. And a few weeks ago, I read a media statement that $25 million had been awarded to 19 health and research organisations, including Harry Perkins and Perrin. And the amount awarded to those two was insignificant compared to the amount awarded to Telethon Kids. How do you think we can raise the profile of the organisations that you guys work with to try and even out the playing field so that more funding comes to the really important research work that you guys are trying to do?
Yeah, it's a really great question. And like someone else said, they kind of feel like WA gets left out sometimes. It's the same with the funding as it is with the clinical trials. We don't get as much funding as over East, and even within WA, there's different proportions of funding going to different places. I don't have a good answer. Gina might have a better answer than me to this, because she's kind of more in that space than I am. I think the only thing that, from my experience, is that there's so much good research that really is truly fundable, and there isn't enough money to fund everything that actually should be funded. So a lot of really great research goes unfunded all the time because there just isn't enough money invested at the very first step. But Gina can probably answer the question more specifically about the geographic distribution.
So the major funding source for medical research is the NHMRC, which is part of the federal health budget. And over the years, the funding coming to WA has declined steadily. So we get about 2.5% to 3%. In a good year, 5% of the national funding. And we're 10% of the population and we pay our taxes. And I think part of the reason for that decline is that there's been a chronic underinvestment by our state government in medical research for decades. And even though our state economy is doing incredibly well compared to just about everywhere else in the entire world, they're still not really investing in medical research. So we now have the Future Health, what's it called?
Research, medical
The furry one.
the Future Fund.
The State Future Health Research Innovation Fund. And I think when they announced it, it was $40 million. And that sounds like a lot of money. But the other state governments are investing orders of magnitude more money than that. So hundreds of millions of dollars for decades. So the state government's $40 million is a drop in the ocean compared to what we actually need. So the more that the electorate can say to the state government, we want some of that money put into other industries, the better. And the state government keep talking about diversifying the economy, but then they go really small with the Yeah, they think big and then they take a zero off the end. So we need them to leave the zeros on the end of new kind of initiatives. Peter Klinken gave a really good talk at the Harry Perkins Oration last year about what we actually need, and not just medical research. Like I was like, go WA, you know, all of the rare earths in the ground here and everything we have going for us here in terms of robotics, and you know, more NASA people come here to train than anywhere else because there's more robotics in the mines than anywhere else in the world type thing. There's so much going for us. We just need the government to think bold rather than thinking small. So yeah, anything you guys can do to help, be great.
Yeah, that's just another bit of advocacy add to the list. More letters. I mean, I personally also feel like we have enough billionaires in this state that there should be a requirement that once you hit a certain X number of dollars, there should be a compulsory donation or investment into philanthropy and research. And, you know, any more questions? Oh. We might make this the last one, only because we're ticking over on time, and I know these ladies, I'm sure, want to get home to their families.
With the when drugs do become available, I know there's, in my case, there's I think it's called labetamide. I don't know how you say it, drug. It's in its third stage. So does the FDA approve it first, and then the TGA have to approve the FDA from there? No.
I think you might have a better answer than me to that one. That's not my area, so I actually don't know the answer to that question. I'm very happy for someone else to take the floor.
Thanks, Piper. No, every country has its own equivalent of the TGA. So as most clinical trials and research is conducted in the US and in Europe, that's where the drugs typically get approved first. Australia typically is between 2 and 5 years behind any approval overseas. The pharmaceutical company has to want to come and invest in Australia, and they don't always want to do that because the money that our PBS or MBS gives back to the pharmaceutical companies, you know, when we go to a pharmacy and we fill a script, it's about $20. The cost of the drug is obviously way more than that. But what the Australian government is willing to pay to a lot of the pharmaceutical companies is less here in Australia than what they could get overseas. So there doesn't seem to be much of an incentive for a lot of pharma to come to Australia because they're not going to get as much money for their drug. When you apply that to rare diseases, where we're such a small population as a whole, a lot of the investment will go to drugs that can treat a huge section of the population, like cancer drugs and diabetes. So no, the pharmaceutical company has to apply here for the TGA. Once the TGA says that yes, it's safe and effective for that drug to be used in Australia, the next step is then going through what's called health technology assessment, where the government tries to determine whether they're going to put it onto the PBS list or the MBS list so that we just pay a fraction of the cost rather than the full cost. And that process in Australia is under review because it's incredibly lengthy. There is a lot of work to fast track that process so that we can catch up to a lot of the other countries that have these drugs that, you know, developed here, but we can't access them. So I'm hoping that, I think it's improving, but there's still a lot of work to do in that. So, no.
Sure. So just one little thing. When you showed the, more about CRISPR, was that CRISPR that you were showing up there?
I didn't show CRISPR. It's similar. The CRISPR will target the DNA, and the therapies that I showed target the RNA. But I can answer a question about CRISPR. I do work with CRISPR.
I'm just interested in the technology of it. Has it got, the actual technology has got a long way to go.
Yeah, that's right.
And it's very interesting.
It is, yeah. As someone else in the room touched on earlier, it's kind of like we actually have kind of the power, the tools exist that we could go and just change a single base in the DNA, a single letter back to the other letter. And, you know, we find that we can do it in the laboratory, in a dish, in some of these cells. We can make changes. The technology is not at a point where it's efficient enough that we can do it with a high enough, in a whole dish of cells, I might get 10 cells that have the change that I want and 10 others that have a change I don't want. And so there's a long way to go in terms of assessing the safety to make sure that you're making the change that you want at a high enough level and you're not making other changes, as well as a whole host of other issues in terms of delivering it to the cells and when do you treat. It's kind of, I think probably CAR T therapy, which is a treatment for cancer, is one where this is being applied and used, but in that case what they do is they take the cells out of the body, make the change out of the body in culture, screen them really, really thoroughly to make sure they've only made the change they want, select for the ones that have the change they want, and then put those back in the body, which is a bit different than, say, a muscle, which is 40% of your body mass. It's challenging to deliver treatments to enough of a muscle, or even just a diaphragm. Given its location, it's a smaller muscle, but you know, there's more challenges with muscle than there are for things like blood. So those other kind of areas are leading the way.
Some lady crystal fly side or something like that.
There's actually some really, I am not really on top of it, but I know there are some exciting things happening in the eye space because there's the research centre located here, Lion's Eye, sorry. I was like, I know it's, what's it called? Yeah, so, yeah.
Thank you so much. I just did a quick little Google. What do you call it? What's your PubMed? I did a PubMed on the Google. And just thinking about, if you can remember back to December 2019 when COVID hit, to get the COVID vaccine to being delivered to humans was literally a year, but the whole world was working on it. So trillions of dollars was being spent on it, and thousands of brilliant minds were being spent collectively to develop therapies. So, and that took a year. So and the whole world had to act on that. So I guess it just brings it back to relative to individual conditions and the size of those conditions or the number of the population. But I'm really excited because, you know, we have brilliant minds here who are doing amazing stuff, 2 of them in the room with us. But also, and one thing that hasn't been touched on, I think there is great advances in AI technologies and what AI can bring to research in analysing a lot of the data that is coming out that would have taken, you know, weeks or months. I think there is opportunity down the line for some analysis progress speeding up. But I think there's so many more questions and there's so many more topics. Tonight's done. So I just want to thank you both very much for coming along and presenting. We do have some beautiful little flowers for you. Thanks to Piper. Just to say a massive big thank you. So there we go. You can look at them and remember us and be reminded that you're coming back. And thank you to everybody here for coming along tonight. We can't host these without an audience, so it's really important that our community come along. We have left some little surveys on the table. Please fill those in because those will help us in preparing for future events and what worked well, what didn't work well, what else you'd like to see. Anything that was right or wrong for today. I just also want to say a massive thanks to Dimitri at the back of the room who is videoing for us tonight. We have an amazing videography partnership with Clip Media, and they are producing our videos so that we can share with the rest of our community. So thank you, and pass on to Stu as well. Thank you so much. Who have I missed, Piper? I have probably missed Piper and Josh. Well, I haven't missed them, but a massive thanks. Piper coordinated the whole of this evening, and so a big shout out to you. Thank you so much. These evenings have actually been driven by community feedback, and also our research advisory committee from a board level said, oh, it would be great to do more research updates. And so thank you so much. Josh, thanks for coming along tonight and being our guest welcomer. And yes, reach out to us if you have questions. Lovely to see you all. Drive safely home. That's it? That's it. Awesome. Thank you.